أهلا بيكم في دروس الأحياء للصف الثاني ثانوي لغات. Today's lecture will continue our discussion about excretion, and this time it's in plants. So let's begin. Excretion in plants, the rate of which is much lower rate, because the metabolism is, or the catabolism is much lower rate than that of animals of equal weights. It's very slow. The metabolic waste, accumulation of metabolic waste is very slow. And also the plants reuse the catabolic wastes, like for example the carbon dioxide and water produced by respiration. It's utilized in photosynthesis, and all of you know that. Also the plant utilizes the nitrogenous wastes in the synthesis of the required proteins. Since it depends on its metabolism on carbohydrates, metabolism wastes are less toxic than those resulting from protein metabolism. Also the metabolic wastes, like organic salts and acids, are stored in the form of insoluble crystals in the cells of plants, mainly in the cytoplasm or in vacuoles. This is how plants utilize their excretion function by different means, like you studied so far. Many plants also has get rid of carbon dioxide and some mineral salts through the roots, okay, and also through the stems or through the leaves, which lead accumulation of elements inside it, and eventually it's shed off. Carbon dioxide that results from respiration and oxygen obtained from photosynthesis excreted through the stomata, pores, means pores on the leaf, by diffusion or by the process of guttation. Guttation اللي هو الندى، قطرات الندى اللي بتتكون، بس مش من الجو اللي بيكونها، لا، النبات نفسه بيفرز المية الزايدة اللي عنده من خلال قنوات بتفتح على السطح ومفتوحة على طول في النهار والليل يعني. So they are water stoma called hydrathodes. دي البورز اللي منها بيخرج المية، hydathode. Guttation water contains other substances which can therefore be deposited. So the guttation is important means of excretion by which the plant utilizes to get rid of excess water. Water also passes in the form of vapor in the intercellular spaces of the mesophyll layer. Lots of intercellular spaces are present between the cells there, and water vapor evaporates through it to the stomata of the leaf. Actually, evaporation of water is over the total surface of the plant. This is this loss of water is called transpiration. 90% of the total lost through stomata on the leaves. So this is called stomatal transpiration. If it occurred through a cuticle of a plant, so it's called cuticular transpiration. If it occurred through lenticels, lenticels are pores in the barks of plants. ثقوب في اللحاء بتاع النباتات وليها قنوات ممتدة لجوه ومفتوحة على طول طبعا. ده اسمه lenticular transpiration. Okay. So we have different kinds of transpiration, stomatal transpiration, cuticular transpiration, and lenticular transpiration. All these leads to the getting rid of excess water in the plant. And the majority of the loss comes through stomatal transpiration, as you've studied here. Now, there is an experiment to illustrate transpiration. A potted leafy plant and cover it with paper saturated with paraffin. Cover the whole thing with glass bill jar, and put it on a glass sheet, and observe what will happen during hours. There will be tiny droplets of water appear on the inside of the bill jar. This means that water is formed and accumulate and become bigger in size and run downwards on the wall of the bill jar. Conclusion: air inside the bill jar receives water vapor from the plant. This is how the plant is getting rid of its excess water. And if you examine this against copper sulfate, it will turn blue. This means that the formed molecule is water molecule. So we can conclude that water passes from the exported parts of the plant to the surrounding air. Okay, this is what this experiment concludes. Importance of transpiration for the plant: to control or decrease the plant temperature, and to help in the ascent of water and salts from the soil. Now, all the metabolic reactions occurring inside a plant, the energy absorbed may be turned into heat inside the leaf tissue, and especially in hot weather, this will be harmful to the plant. So it must use transpiration as means of decreasing the temperature of the plant. This is one thing. Another thing is that the root cells contain the sap, which is higher concentration than the surrounding soil. That's why solutes is being pulled in by means of osmosis. Okay, this is the root pressure, and it uses the osmosis until it reaches the xylem network, and then ascends through the xylem vessels and to the leaf veins. This happens. This comes to an end as it reaches the mesophyll cell sap. Why? Because the decreasing ability of these cells to pull up more water. Okay. There are intercellular spaces full of air between the mesophyll cells. As a result of continuous loss of water from the mesophyll cells, their concentration increases. This leads to creation of a pull force, pulls water upwards. Okay, and this explains the transpiration cycle. Transpiration aids in pulling up fluid from the root through the xylem network. Osmotic pressure, as we discussed before, was not enough for trees that reaching high up to 125 meters. Therefore, we discussed the theories of how the sap ascends to these high altitudes, like 125 meters, and the most acceptable theory was the theory of adhesion and cohesion theory. Remember that? The one that talked about the molecules of water and the forces pulling up the column of water until it reaches the intercellular spaces in the mesophyll layer of the leaf. An experiment to show that water ascends through the xylem a test tube, fill it with eosine, and bring a leaf, a plant with roots, and put it inside the tube and cover it with cotton and wait for hours and observe what happens. Observation that the leaf petioles will change to pink, as you can see here, means that the roots took up the eosine in the solution, and through the xylem vessels, xylem network, it reached the leaves. So this is the observation. Then you're asked to cut a thin section and examine under the microscope. What will you observe? You will observe that xylem tissue only is stained by eosine. This only stained the xylem tissue. Conclusion: pink coloration of the petioles and petal veins indicates that eosine solution reaches these organs. The experiment shows that, first of all, water is absorbed by the roots, and we discussed that earlier. Remember what we talked about, imbibition and so forth, and the high concentration inside the root cells. And water ascends through the xylem tissue of the stem to the leaves, and this is proven by this experiment. Okay, one last experiment to show the ascent of water by transpiration. Well, you're asked to fill a small beaker with mercury, bring a tube filled with water, and immerse the lower end of it, invert it and immerse it in mercury. And at the top, you attach a rubber tube with a leafy twig that takes, that's dipped in the water. What will you observe? That the mercury rises in the tube. Why? To fill in the space left by the transpiration of fluid from the leaflets of the plant. Because the transpiration occurred, this led to reduce the amount of water in the tube. So to fill in the missing part, the part that transpirated, the mercury rose up in the tube to fill in its place. Conclusion: the rise of mercury is due to the transpiration pull, where the leafy twig loses water by evaporation. This indicates that water loss by transpiration generates a pull to raise the water upwards. This concludes our discussion of excretion by concluding excretion in plants. I hope you got the maximum benefit from this lecture, and thank you very much for watching.